Welcome everybody to this Christopher Pankhurst Institute seminar. We're focusing on translating emerging technologies for health, ethical and legal considerations. It's in the sort of responsible and inclusive innovation theme, which is sort of partly born out of work on responsibility. Work from the likes of Sheila Jasanoff, who was writing about co-production in 2004, where social order and knowledge emerge together. And also from writers like Jack Stilgoe and Richard Owen, who were writing about responsible innovation, or from science and society to science with and for society. And they worked on things like the area framework for EPSRC, which is to anticipate, reflect, engage and act. So trying to move some of the responsibility for anticipating and engaging stakeholders in innovation upstream and into the earlier in science and innovation. So some of this is rather than the idea of selling emerging technologies to publics who are assumed to be sceptical of science due to sort of lack of technical expertise. That stakeholder and public steering of research and innovation agendas is suggested to support what Novotny and others would call a socially robust technoscience in its emergence. So one claim is that publics are not ready-made, but are created through engagement and communication practices. And so to attend to who is included in science policy, what for, and with what kinds of power, some of the kinds of questions for consideration. So the format of this seminar is we're going to have 3 10-minute talks, and then we'll have some discussions and questions at the end. So we've got 3 talks from 5 scholars at the Centre for Social Ethics and Policy, which is known as CSEP, and it's located in the law school, has a range of scholars and researchers who are contributing to philosophy, law, bioethics, healthcare ethics, and policy. With foci areas including reproductive ethics, end-of-life care and assisted dying, transgender identities in children, regulatory considerations for medicine and healthcare, and emerging technologies like stem cells, cloning, transplantation, and so on. So what I'd first like to do is invite our first 2 speakers, who are going to be Research Fellow in Bioethics Jonathan Lewis and Professor in Bioethics Soren Holm, to give their talk, which is addressing regulatory gaps and uncertainties for use and emerging technologies in embryo models.
So this comes from research that we're doing within a Horizon 2020 research program on organoids and essentially things that you can grow from stem cells. And we'll here focus on regulatory gaps and uncertainties for embryo models that you can, from stem cells, grow things that are very much like embryos. And at least in certain animals, you can grow things that are embryos in that they can be implanted and give rise to live births. Whether they are exactly like embryos, we don't know in those animals yet, because we don't know whether they sort of grow to normal adulthood and get old in the normal way. But at least everyone would, in these animals, count them as embryos. Now in humans, we can do various embryo models. So the question is, how should we conceptualize these things, and does that matter to how we should regulate them? One approach to this is to ask, what are these entities? More specifically, because that's the way we have regulated the reproduction use, are they embryos? So one thing that we've been looking at in the project, and we'll talk about today, is whether this is likely to be a productive approach. This is the right question to answer, or ask and answer. And obviously, the question relies on the implicit assumption that if we know this, the answer, we also know how to regulate but that's not obvious that that's true. At the moment, we regulate embryos in reproduction, even though we have no consensus about how they should be regulated, what they are, sort of what more status they have. So the uncertainties, lack of consensus about embryos have not so far stopped the regulation of embryo research. So the whole aim of the project is for a range of organoids to answer some conceptual questions, some epistemological questions. So what are they? How do we find out what they are? And then the regulatory questions. So what is an embryo? Well, the problem here is that we have many possible answers. This is the non-exhaustive list. It might be something which we define biologically. It might be something we define theologically. It might be something we define by not what it is now, but what it can become, either biologically or in terms of other types of potential. And it's very unlikely that we can reach consensus at this level, that we will ever agree on what an embryo actually is. So that then raises a further question about epistemological uncertainty. Now, we know that researchers are aiming at a model that is as close as possible to the real thing. At least some researchers have that as an aim. And the first important thing to note, which is sometimes forgotten in the public debate, is that just because researchers say that this is what they want to do doesn't mean that they can do it, or that, and even less that they have done it. So if you want to develop a model of a blastocyst, a specific stage of an embryo, it doesn't mean that you have developed that model. It just means that that's your goal. And it's also important to note that at least some of the answers to the question about what an embryo is means that these things could never become embryos because they have a different genesis. So they don't start out in the ways that embryos start out. Now, in animal research, we try to find out whether a blastoid is a good model of blastocyst by implanting it into the womb of a receptive female animal. Now, for rather obvious research ethics reasons, we cannot do that in humans, even if it was legal, which it is not. So for these human models, we might end up with having sort of a residue of epistemological uncertainty about, are they still just models, or are they now the real thing? Because we can't do the research that could prove that they were the real thing, because that would require the complete development of this entity into an adult human being, which we can't do. So we have the issues with regulatory uncertainty. What is the proper aim of the regulation of these embryo models? What is it that we want to regulate? Does it the answer to this question depends on knowing sort of what they are, either ontologically or epistemologically, and can we regulate on the basis of what is possibly unresolvable uncertainty? It may well be that we will never be able to say whether the particular entity that has been created in a particular lab is an embryo or not.
Hand over.
Yes.
Okay. So regulatory uncertainty. I'm just going to go back to this slide. So Soren came up with these Sort of three questions that really would be reasonable for any regulator to consider in order to capture these embryo models. So if we start with these questions, and let's go back to first principles in terms of how we can answer them. So broadly, what everyone can agree on in this area of research is the recommendation that was made by the International Society for Stem Cell Research in 2016. And that recommendation was that no human embryo may be cultured outside the body beyond 14 days after fertilization. So because embryo models aren't covered by current regulations governing embryo research, sort of the aim of regulating the research of embryo models should be to capture these models within a framework for embryo research. And going back to first principles, to ensure that no human embryo is cultured outside the body beyond 14 days after fertilization. So in other words, we need a framework to cover those instances where the regulatory distinction between an embryo model and a real human embryo disappear. So, second question, does this answer to the first question depend directly on the ontological and epistemological certainties, resolving them? Well, notably, it doesn't necessarily require that we resolve either the conceptual or the epistemological uncertainties. We don't need to determine what a human embryo in fact is, i.e. biologically, naturalistically, metaphysically. And we don't need to know whether the blastoid we are developing really is a human embryo proper. So rather, instead, what we need is a normative definition of a human embryo that would include some possible embryos, i.e. those models where, as I say, the normative distinctions with an embryo disappear. So these are the third question, can we regulate on the basis of this possibly unresolvable uncertainty? Well, in August 23 this year, a group of biologists, bioengineers, stem cell researchers, geneticists, and medical ethicists attempted to answer that question. And they redefined the embryo so as to make it, at least in principle, possible to regulate embryo model research in the face of these conceptual and epistemological uncertainties. And they went for this definition here: a group of human cells supported by external elements fulfilling extra embryonic and uterine functions that combined have the potential to develop into a fetus. Now again, they're not saying that this is what an embryo in fact is, nor do they know that this is what an embryo is. In fact, they acknowledge that this definition diverges in many ways from those offered by biologists. And furthermore, they recognize that the current formation of embryo models requires the use of cells that rapidly accumulate abnormalities. So it's very, very, very unlikely, if not impossible, to form a fetus. So they acknowledge that they do not know whether embryo models are or ever will be more than models. Rather, what they're doing is proposing a normative definition that can function to offer protections for some possible embryo models in ways similar to those afforded to embryos under current regulations for human embryo research. Leads me to sort of the final slide. A human embryo model could be legally considered to as similar to human embryo if it is shown to have a potential to form a fetus. That's Riveron and colleagues' point. So what they're saying is once it passes this, what they call this tipping point, it will become fully entitled as an embryo, regardless of how it's formed. And they propose two, as it were, Turing tests to identify whether a tipping point has been reached. The first test would be a given human embryo model shows the potential to produce the complete embryo in vitro as it normally forms up to a certain stage of development. And the reason why this is a normative definition is because, importantly, individual jurisdictions would have the abilities to set the time limit for the test according to their local values and other legislation that operates within the jurisdiction. So it's a normative definition. They're not saying we'll take it up to this point, we should take it up to this point, we should take it up to this point. It is for the individual jurisdiction to determine. And the second test would be an embryo model formed by the SEM methodology, has the potential to produce live and fertile animals in multiple species, particularly those closely related to humans, such as pigs and non-human primates. And they're saying basically the choice of species should represent a trade-off between their entitlement to protection, so more protection further up the scale, and a development similar to that of humans. And again, what they're saying, this is a normative test because they're saying this balance should be evaluated in the context of local values. Yeah. Now, the regulatory definition they propose is no means perfect. There's still uncertainties underlying it. For instance, precisely what fetal stage their definition refers to is unclear. And also, what counts as a genuine embryo model rather than simply a cell or tissue culture is also unclear. But that said, what it does illustrate is a possibility for regulators to respond to new and emerging biotechnologies, despite there being a lack of agreement or certainty as to what these entities are and limitations on what experts know about them.
Lovely, thank you very much for that talk on regulating embryo models. Does anybody have any clarification questions? Because I was going to just move swiftly on to the next presentation, if there are none. So the next presentation, is looking at the GP Data Trust project about empowering patients and engendering trust in healthcare data sharing. With Catherine Bowden and Sarah Devaney.
Yes, thanks very much for the invitation and for your time today. Kath and I are going to talk briefly about our work on GP data and the potential for data trusts to be used to share that data for research purposes. We're going to talk briefly about some of the work that we did with patients to find out their views about their own GP data and its use in research. So Kath is going to provide you with some details of what we found there, and then we're going to just give a very brief account of the work that we're leading into now to extend that to another proof of concept study. So I'll hand over to Kath.
Okay, so this was the GP Data Trust study that we ran last year, involving data collection to explore how a data trust might work in the context of health data sharing. And this is a bit of the background, which you might be aware of, to the study. So in May 2021, NHS Digital announced that they would be collecting patient primary care or GP data to be used in a non-identifying form for planning and research in their GP Data for Planning and Research, or GPDPR programme. And this was some of the background, the response to it. So it quickly gained the far from neutral label of the NHS data grab, and over 1 million people opted out of the programme in 1 month, which prompted NHS Digital to pause, to take time to consider people's concerns. So the aim of our project was to explore why people had opted out of sharing their GP data and whether a data trust could address some of those concerns. So these were the activities we undertook So we had 2 online surveys, which ran for about 2 months. And we had a fantastic response with 184 patients and 86 GPs. And we did 18 qualitative interviews as well, with 10 with patients who'd indicated that they had opted out, and 6 stakeholders. So people, campaign groups and people working in the area looking at these solutions, and 2 GPs and practice managers. And we also ran 2 focus group sessions in partnership with the Patients Association, with 10 to 11 participants in each, with a mixture of views. So as part of those focus groups, we had an illustrator who produced this lovely graphic for explaining our project to people. So you can see we have the NHS data, GP data, going to NHS Digital for then to be accessed by various researching institutions. And overseeing that, in the corner you can see that there would be a data trust governing the access to that data. So trustees who owed legal duties to the beneficiaries, who are the people who had contributed their data to the trust, and that would be overseen by the courts to check that the trust was being run appropriately. And there could be multiple trusts as well, based on different locations, different purposes, different terms on which the data would be shared. But the key to the trust is the nature of the fiduciary duty. So rather than just a contractual duty to fulfil terms, you're looking at a duty to act in the best interests of the beneficiaries and with undivided loyalty. So it brings in some more ethical concepts. So this is another illustration from our scribe, and this is summarising one of the discussions from the focus groups. So I don't know if you can see the pointer up there, but so you can see here that there were concerns about the communication, that there should have been more information provided, and that patients were made to feel vulnerable and disrespected by the way it was handled. There were concerns about the data being sold to commercial companies and who would benefit from that. We also had a lot of concerns expressed about the accuracy of the records as well, because when people have seen their records, they spotted mistakes. And overall, down here, we can see that people wanted to know how their data was being used and who by, and have a control over that. They wanted it to be their decision. So this is the analysis of the data which we're doing at the moment. And the main theme coming out of that is this relationship between the ownership of the data and the trust that it would be handled appropriately. So the lack of information, the opt-out nature of the decision, and the short time frame that people were given made them feel that it wasn't being treated as their data, and that meant that they didn't trust it would be handled appropriately, and so they opted out to assert ownership of the data. And that's shown in this one of the quotes from our participants. So they tried to make it that the data belonged to the NHS, and it's not, it's mine. The only person who has a say about that is me. So at the moment, what we're doing is a bit more analysis on what this concept of my data means to our patients, and what that tells us about the type of control they want to have over the data, who they see the value of the data belonging to, the effect of anonymisation on the concept of my data, and how patients could be harmed by the uses of their data. And the next bit of our project is going to look at how the way data is managed can reflect this concept of my data.
So in terms of next steps then, alongside this ongoing analysis, we're developing the proof of concept stage of the work at a regional, Greater Manchester level. So we're very keen to make sure that as we take this work forward, we have at the core of our work the knowledge that people do want to have a say and control over their health data. That's very much within a general willingness to share data for beneficial health research uses, but that does come with a caveat about having some input over that. So people also told us that they want this trusted legal and regulatory structure, and also that they're concerned that there's a secure technological environment within which that is all happening. So we're fortunate to be affiliating to regional innovation in the use of health data for research, and we want to use that to advise regional and national developments in using health data and engaging in health data management. So the regional activities that we're affiliating with relate to the Greater Manchester Integrated Care System. These are partnerships that bring together NHS organisations, also local authorities and others to take collective responsibility for planning, to improve health and reduce inequalities across regions. NHS England made an initial wave of funding awards to various different regions, of which Greater Manchester was one, to develop secure data environments for research and development in connection with the integrated care systems. The Greater Manchester work will be building on the excellent work of the Greater Manchester Care Record, which is a drawing together of the region's primary care data. So there's this matching there of the primary care data of our focus and the work that was undertaken in the care record. During the pandemic, this was really expedited, the gathering together of primary care data, dealing with requests from researchers to use that data for beneficial research in a COVID setting, and that's now expanding more generally. And so there's this national plan that there'll be an interoperable network of subnational secure data environments to enable patient data to be safely and securely accessed by researchers. And our affiliation to that work brings in everything that we know from patients about the say that they want to have about how their data is used and the interactions with them in terms of getting permissions, obtaining preferences and so on that goes along with that. So we've received a bit of extra funding for GPDT2, which will allow us first of all to undertake some technical work. I think James Cunningham, our tech expert, might be on this call. Can't see you, James, if you are there, but James is looking at developing a patient-facing dashboard that will enable patients on their phones or whatever to be able to indicate what preferences they have about how they share the data. So if they're told that there's some work that's potentially going to be undertaken in relation to cancer or diabetes or something like that, do they give their permission? And so this isn't the same sort of system that we have been seeing where it's a one choice for all time, opt in or opt out. It's more granular than that. And so we're liaising with the Greater Manchester Group to develop this system to be integrated with their work on the secure data environment generally. We've got a legal and commercial strand to this to explore the detail around the type of structures. So we've been exploring whether a trust could work, and there's lots of support for the involvement of a trusted intermediary there. The Greater Manchester Care Record governance probably might not have thought of itself of this as this, but it is very trust-like in its structure, or has been. So the involvement of specialists with different backgrounds, technical, clinical, statistical, patient involvement and so on. Lots of these really excellent elements that patients tell us they want to have there is there, but it's building in this communication between patients and that governance structure with those added guarantees that a trust might bring that we're going to explore. And then we'll be really keen to make sure that we're disseminating all of this at regional and national levels. One element that did appear in our initial pitch to the funder was continued work with patient and public groups. We haven't abandoned that at all. The knowledge that we've gained already drives all of our questions, but we are applying for some additional knowledge transfer and engagement funding to make sure that we are able to focus group and test the development of the dashboard with patients as well. So we won't be neglecting to get advice from patients and public participants in relation to the development and use of that. So those are our plans based on our first stage findings, and I think we'll probably be doing questions after the next
That's all right.
presentation. Won't we? Yes, that's great for us. Thank you.
Right. Right, thank you very much. And I'm now going to move online for Lucy Frith, who's a reader in bioethics, and her presentation is going to be on direct-to-consumer genetic testing and the ethical and social issues to do with gamete and gamete donor testing.
Okay, well, thank you very much for the invitation, and I'm really sorry I can't be there in person, so I don't know what you can see of me, but I'm going to talk about a ESRC funded project that is looking specifically at direct-to-consumer genetic testing and the implications for egg, sperm and embryo donation. So this project, as I said, funded by the ESRC, and this is the project team. I think Caroline is in the audience as well, so she'll be able to correct me if I say anything wrong. So the broad aims of this is to understand the regulatory, social and ethical implications of direct-to-consumer genetic testing in relation to donor conception. So we're interested in both how people are using this technology, but also how we should think about potentially regulating it, and not necessarily saying we need more regulation, but exploring if we do and in what context. So what we're doing is we've conducted interviews with donors, parents through donor conception, donor conceived adults who have engaged with or who have been affected by online DNA testing. We've also held workshops to discuss our interview findings and their implications, and we're planning to co-develop some support resources. The other strand of the work, which Caroline is leading on, is reviewing direct-to-consumer genetic testing company terms, conditions and laws and policies around the management of information about donor conception. And we've chosen to do this in 4 key jurisdictions: the UK, Sweden, the Netherlands and Australia. And Caroline's carried out some interviews with experts, and we're convening a legal regulatory roundtable in January to try and pull together our findings and seeing if there's any regulatory implications, both for how these companies themselves might be regulated, but also in terms of the regulation of gamete donation in the UK, if any changes need to be made in that regard. So what I'll do in this short presentation is take you through some of the key findings from the interviews. And then I think the sort of legal regulatory bit is something we can do for another time. So we interviewed 18 donors, 25 donor conceived people aged over 16, 20 parents through donor conception and 2 relatives of donors. And like most qualitative studies in the area of fertility, fathers, men are usually underrepresented. Also parents following sudden disclosure, clearly donors who don't want to be traced, and people from minoritized backgrounds. So we were interested in how people are actually using direct-to-consumer genetic testing. So that might be Ancestry, 23andMe, the places where you see those adverts around Christmas, where you find your history, find your relatives, and so on. And there was a number of ways that this newish technology impacts on the donor-conceived community. And that's just a summary of the possible uses, and I'll talk about those in a bit more detail. Okay, so one obvious and important way that direct-to-consumer genetic testing is impacting on the donor-conceived community is that people are actually finding out they're donor-conceived by taking these tests. So they may take them as a response to those adverts, want to find out where their family comes from, or Victorian ancestors, and then discovers that they're not biologically related to usually their father. So this may be that they have suspicions that they are donor conceived and use it in this way, or it might happen out the blue. And these sort of revelations, if you like, can be sudden or they can be prolonged. They might get some results that look strange, as in, why am I matching with lots of people who I don't know, lots of sort of extended family. And of course, there's the psychological and social impact on this. And in this and other research, we found that donor-conceived people's parents' reactions are important to how they handle this. So one of our cases is the case of John, who was born before the 1990 Act that governs this area in the UK came into force. And he sent off for a test for his wife, but by mistake, they sent him another one. And they thought it would be fun to get the results and show them on a projector. And the results identified his biological father, who was not the man he thought was his biological father. And his donor had put himself on the Ancestry site to make himself known. So John was shocked, but has made contact with his donor and has had a positive experience. But he hasn't told his parents he knows or told his younger sister. So another way that people are using this technology is for searching and finding donor relationships, relatives, sorry. So people may be looking for donors or donor siblings. And often direct-to-consumer genetic testing is preferred over available official routes. So for instance, in the UK, where there's the HFEA, if you're in certain situations, you can find out information about your donor. People are preferring these because they sometimes say they feel more in charge, more in control, and so on. And of course, the donor does not need to be registered themselves to be identified. So if a donor's close relation, a sibling, for example, is on, then it is possible to trace family relationships that way. Often contact is positive, but sometimes matchers won't want to engage. And of course, it must be remembered that not all donor-conceived people will find their donor. Another way people are using it is donor-conceived people and their parents are seeking connections beyond the donor. So this is something that this technology enables, where the HFEA is very concerned about donors, possibly donor siblings, but that kind of triangle. Of course, you can make contact with the mother of your donor, for instance, and we've had cases where people have developed quite good relationships with their donor conception grandparents, if you like. And another way we found is that gamete donors are using it as a way of making themselves available for donor-conceived people who have been born through their donation. So they may have donated before any records were kept by the HFEA, or they may have donated in the period between 1991 and 2005, when donation was anonymous. So this is an interesting way that some of the donors have said to us they feel some obligation. They'd like to be available to any donor-conceived people. And one thing some of them hadn't considered is that the donor-conceived person may not be aware that they were donor-conceived. So that's a sort of complication they hadn't considered. A further way is donor-conceived parents using this technology to make connections between families. So it may be used to build extended families when people's children are young. Some people place a high value on creating sibling-style relationships between different donor-conceived children. And sometimes mothers through egg donation feel a sense of connection with the donor. And there's some tensions that were perceived here. For instance, a child's ability to make their own choices about connections and the donor's right to privacy, at least till the children reached the age of majority. So I'll just conclude with some challenges that this technology brings up. So the first challenge is being an intermediary or a gatekeeper. So relatives of donors and donor-conceived people involved and others may find themselves holding information about other people and being gatekeepers of that information. So for instance, if a donor-conceived person doesn't know they're donor-conceived and contacts you because they've got some connections with your family, you know, do you tell them they're donor-conceived? You know, you are in a position of having this information, and secrets are very common in this area. So it's felt that it was putting a burden on some people as being sort of holders of this information. The second challenge is family dynamics after late disclosure. This research and some research I've done previously has shown that when people find out they're donor conceived later in life, you know, it can be any age from you know, up to 60, there's often a lot of questioning that goes on, impacts of family dynamics, managing people's emotions and so on, and a lot of feelings of people's parents keeping important things from them and creating new pockets of secrecy. So the case of John, where he found out he was donor conceived, but he hadn't talked about it with his parents. So they've not told him, but then he's not telling them that he's found out he was donor conceived. And the final challenge was dilemmas around the early testing of children. So we found quite an interesting division in our respondents in terms of very different views on this. So it is possible to test children from a very early age, but the ethics of doing this were strongly contested. So some of the advantages were gathering information for your children to pass on, connecting with donor siblings in childhood, and removing the pressures of the decision for the donor conceived child. However, other parents thought that you should wait until your child was able to make that decision for themselves. They also felt concerns about sharing their child's genetic data on these sites and worried that finding the donor too early when the donor conceived person was a young child might undermine the relationship with the donor. So that's a bit of a whistle-stop tour of the many issues that we found with this. But if you've got any questions.
Thank you very much, Lucy. And I'd like to thank everybody from CSEP for agreeing to give their presentations. So Nicola, would you mind just checking on, have you got your, you logged in, would you mind just checking if anybody's online asking questions? So, yeah, I'd like to open this up for questions and discussion, really, if anybody has any initial thoughts or anything that you'd like to share. Nicola's just going to check online if there's anything in the chat. Okay. So I suppose that just my initial reflections, a couple of things. One is that we've got lots of different, the three presentations looks at different technologies that are in different stages or phases of translation. So there's sort of Jonathan Soren looking at science and regulation of a kind of new emerging entity. There's the health data and how people are relating to trust to health data institutions and how they control the data, and then looking at something that's already at market and how that's almost at the intersection of two different technologies, one around consumer testing and one around donor conception, and how those two things are intersecting to sort of reconfigure how people relate to one another and their understandings of kin and family and so on. So I think I have a question to all the speakers really, which is, how do you see your research as feeding into or impacting or changing innovation as it's happening, or translation, whichever you would like to say it. So I don't know if any one of you five would like to field that initially.
Well, I can start from the embryo models in the sense that they're very likely always to remain a research tool. Now, a particular way of developing a particular embryo model might well be commercialized, but that's sort of commercialized within the very narrow space of tools for the pharmaceutical industry, broadly conceived. So in that sense, the translation and the translation step is perhaps not as large either, because it's translation and commercialization to other experts. Now, I think what we hope to do is to identify some regulatory gaps and identify, well, it's paid for, our project is paid for by the EU, so in the EU context, identify which particular regulator is the one who ought to do something about this and gives them some options. But then obviously, so when we're talking embryo models, it's so hugely politically sensitive that sort of it is essentially a political question, which of the many options will be chosen.
From our perspective, I suppose there are two elements that we contribute to. One is the governance side. So it's an attempt to ensure that governance of data that's used in health research is informed by the patient voice. At the moment, the governance structures that activity works within tends to assume that health data is a sort of social good and a social resource, which it is, but that shouldn't be at the expense of the loss of voice of those who've made it available, or those that it comes from, because it has a much more important and personal connection to them as individuals than the governance framework says. So it can give researchers the confidence to know that they're using that social good on a kind of more ethical basis. But also, I think in turn that can, if there's more buy-in, if that encourages people to say, yes, I would like to opt in, I would like to choose to do that, it can add to the quality of the data set as well, because you may have a fuller data set if that means that people are just more confident to join it, but also one that as I say, is sort of more ethically founded.
Thanks.
Yeah, maybe if I could just quick, I mean, I think in the area of donor Can people hear me? This is very helpful.
Yeah.
I think in the area that Caroline and I are looking at, you do feel that you're almost doing a kind of post hoc, ameliorating some of the potential consequences of it. So the impact on direct-to-consumer genetic testing for donor conception was probably very far from anyone's thoughts when it was developed and marketed. So I think it's interesting that we probably never do know the full effects of rolling out certain technologies. We have the sort of best laid plans and think about the potential consequences, but there's always things that we may not have foreseen. So I think we need a way of somehow, even when something's been approved and is up and running, to have some continual reassessment of that.
Yeah, thank you, Lucy. Yeah, I think that speaks a little bit to sometimes feeling like you're invited on to something to analyse it when it's already in a long way in its sort of developmental process as well. Neil, do you have
Yeah, I had a question. I think for all speakers, but perhaps mostly for the first two presentations. And I think, well, background is, I think, probably something that you said, sir, in your talk. So I very much think that we tend to understand the world using metaphors. So we see things and we try to understand them by comparing them to things that we already know. And that's certainly also true for new technologies. And I think very often with new technologies, we're struggling to find the right metaphor, actually, to understand what's going on, what the technology really is doing. So I was wondering, well, for the technologies that you discussed, whether we've got the right metaphors or whether we should still seek for better ones.
Well, for the technology we talked about, I don't think we have the right metaphor terminology. And of course, that the alternative ones, we could have talked about synthetic embryos, which some people talk about, but these things which we tend to think is not a good term because they're not synthetic in any sort of meaningful sense. And then sort of lab created or that. And of course, the particular metaphor or term we eventually pick out will focus on some aspects of these entities and will obscure other aspects of them. Of course, we have also here today talked about like as if everyone who is doing this work wants to make embryo models that are as close to embryos as possible. But there are lots of people doing this who do not, sort of their goal is separate. They want to make models that, yes, recreate some parts of embryonic development, but not other parts. And so the landscape itself is also hugely complicated and contested.
It's the same with brain organoids as well. There's lots of debate about how and whether we and in what extent we should regulate organoids or so-called neural organoids because of potential fears or risks of these things attaining some level of consciousness or some level of sentience. And various terms get bandied about. Should we refer to them as brain organoids, mini brains, cerebral organoids, neural organoids? And with each term, there's built-in assumptions about what they're trying to do. When actually, when you speak to probably the people making these things, they don't see them as mini brains. They don't see them as organs. And in fact, they probably don't like the term organoid because immediately the public starts thinking, oh, it's something to do with an organ. When actually the people making it, they're not organs. They're nowhere near it. They're more like a cell tissue line than they are an organ. So to an extent, you have to blame the researchers for coming up with the terms, but how those terms then get propagated in the public domain, that's a question for the media. And I don't think the media often helps, especially with things like which are very politically sensitive, like brain organoids or embryo models.
Yeah, I think it's a very good point. I think there's definitely a power of metaphor in how people understand things, and what the concerns that patients have over their GP data, for example, is people know that patients want to have a say and have control over it. But I think one of the metaphors that has been used to explain that in the past has been a kind of a property metaphor. So seeing it as, well, they want to have a say over it in the same way they have a say over their property. And then that, of course, leads to things like the GP DPR with a one-off decision of an opt-out. You just have my data and that's the end of it. And what we've looked at from our data is whether actually this concept of my data is actually more akin to the metaphor of bodily tissues, and they see it as actually part of who they are and part of what makes them, gives their identity, and then the implications that then has for what types of control and the regulation involved. So I think there is a shift there that can happen that can really help people to understand what patients want and what their view of it is, rather than kind of imposing a pre-made structure on it.
Thanks very much, Catherine. Lucy, did you want to respond to the metaphor question? No, okay. Okay.
No, thank you.
Okay, thank you very much. Well, we've got 1 minute to go, so I think I might draw it to a close. So thank you very much again to Soren, Jonathan, Sarah, Kath, and Lucy for your presentations, and and thank you for the questions and discussion. So.
Thank you.